Good day, this is Professor Will, CBMD PhD, case Tuesday, March 26, 2019. It's 4:46:25 Eastern Standard Time, and this is a short definition on bacteriophage. So let's get started here. Bacteriophage. A bacteriophage, or simply a phage, is any virus that infects bacteria. Any virus name that ends with phage is a bacteriophage. For example, phages that infect cyanobacteria are called cyanophages. Phages and all other viruses exist as obligate parasites, meaning they must infect a host cell for their survival. As parasites, the viral uses that cause infection usually harm the host. Bacteriophages differ from viruses that infect animal or plant cells because a phage must have a mechanism that allows it to traverse bacteria's unique peptidoglycan cell wall. Microbiologists in the late 1800s suspected that some sort of tiny life form attacked bacteria, but these things were invisible in microscopes and passed through filters that caught bacteria. Scientists in laboratories were forced to rely only on their intuition because they had yet to find solid evidence of these mysterious particles. In 1915, the British bacteriologist Frederick Thorpe, 1877-1955, isolated particles from a culture of bacteria that he suspected were not bacteria. The American Society for Microbiology historian Bernard Dixon wrote in 2001, Frederick Thorpe was a brilliant but eccentric man who for his career engaged in splenetic and often unreasonable conflicts with Britain's Medical Research Council. Thorpe's discovery of phages in 1915 came about by growing sheets of bacteria called lawns on agar plates in his laboratory. After incubating the cultures, he occasionally noticed small clear spots in the lawn where bacteria had not grown. In one simple experiment, Thorpe had accomplished two things. He had discovered bacteriophages, and he had invented a laboratory technique that remains important in virus studies today. But because of Thorpe's isolation with the scientific community, his observations were almost unnoticed. 2 years later, the French-Canadian microbiologist Félix de Hierro, 1873 to 1949 at the Pasteur Institute of Paris, made the connection between the clear zones and bacterial lawns called plaques and viruses. Characteristics of bacteriophages. Viruses have evolved into the ultimate parasite. Their bodies have been stripped down to little more than genetic material wrapped inside a protein coat. Viruses lack all other functions for existence as a living thing. Viruses are said to be metabolically inert, meaning they are not truly living because they cannot reproduce on their own. Bacteriophages reproduce by first infecting a bacterial cell, and then taking over a portion of the cell's replication system that makes more phage particles. Bacteriophages occur in any habitat where their host cell live. Their concentration is enormous, up to 10 million per milliliter in the ocean, but their role there has never been fully explained. Electron microscopy show that seawater serves as a bounty of bacteriophages for study, but scientists have found it very difficult to keep the phages active in a laboratory. Virologist scientists specializing in viruses have conducted most of their studies on bacteriophages that have been more cooperative in laboratory experiments. The main phages that they study infect bacteria such as Escherichia coli, Bacillus, and Pseudomonas. Bacteriophages are classified by two features: their morphology, which is their physical shape, and the type of nucleic acid they contain. Bacteriophages take in a variety of shapes that are visible only by electron microscopy. These shapes can be quite distinctive and so act as an identification aid. Nucleic acid analysis provides a more accurate identification system. Viruses belong to either two of main groups: those that contain deoxyribonucleic acid DNA or those that contain ribonucleic acid DNA. Bacteriophages contain one of four forms of nucleic acid: double-stranded DNA, dsDNA, single-stranded DNA, ssDNA, double-stranded RNA, dsRNA, or single-strand RNA, ssRNA. DNA-containing bacteriophages are thought to be more common and are called simply DNA phages. The DNA phages may be further distinguished by the fact that some contain DNA in a linear structure and others carry circular DNA. Bacteriophage morphology means the shape of the virion, the main body of a virus. Some have a head, called a capsid, atop a short tail portion. Capsids are often polyhedral, made up of many surfaces, and when they attach tail first to the outside of the bacteria, they resemble a lunar landing craft. Other bacteriophages are shaped like long, thin string beans. Bacteriophages tend to be symmetrical and 23 to 32 nanometers in diameter. Several types of bacteriophages have an outer covering called a lipid envelope, made of fat-like compound. Bacteriophages infect bacteria by injecting their nucleic acid into a bacterial cell. Bacteriophages consisting of a capsid and a tail require both pieces in order to do this. Tailless bacteriophages however, have their own means of infecting bacteria similar to virus infection of plants and animals. Bacteriophage infection. Bacteriophages infect cells, reproduce inside them, and then burst out of each infected cell in a process called lysis. Once they burst free, they find other bacteria to infect. This entire process from infection to the next infection is called a lytic cycle. The T2 phage that infects E. coli releases about 100 new bacteriophages when a single E. coli cell lyses. They follow the same steps in infecting bacteria as other viruses used in plants and animals. The 5 steps are landing, attachment, tail contraction, penetration, and DNA or RNA injection. Bacteriophages are used as models in virus research. A large portion of the information on all virus infections has been obtained from the bacteriophage lytic cycle. Details of the 5 steps of the lytic cycle are summarized in the following. DNA phages and RNA phages both carry out this sequence. Landing. Fibrous arms extend from the virion and connect with specific sites on the outside of bacteria, such as cell wall components, flagella or villi. Attachment or absorption. The tail binds with the receptor site. Tail contraction. The tail reconfigures so that an inner tube binds to the receptor. Penetration. Phage enzymes help the tube burrow through the cell wall and make a pore through the cell membrane. Injection. The phage injects its DNA or RNA into the cell cytoplasm. Bacteriophages lacking a tail connect with the bacterial cell using alternate structure. For instance, played a role in the way viruses have evolved along with bacteria and with higher plants and animals. Good day, this is Professor Will, CBMD, PhD. Today is Tuesday, March 26, 2019, and this concludes my reading on bacteriophages. Hope you enjoyed listening. Have a great day.